Welcome and thank you all for standing by. All lines will be on a listen-only mode for the duration of today's call. This conference is being recorded. If you have any objections, you may disconnect at this time. I'd now like to turn the call over to Mr. Ben Beard. Thank you, sir. You may begin.
Thank you. This is Dr. Ben Beard from CDC, the Bacterial Diseases Branch in the Division of Vector-Borne Diseases, and I want to welcome everyone to our annual HHS Working Group on Lyme and other tick-borne diseases webinar that we do. And the topic this year is trends in tick-borne diseases in the United States. And so to begin with, I'd just like to emphasize that the views expressed in this webinar are those of the participants, and they do not reflect the official policy or position either of CDC, the Department of Health and Human Services, or other U.S. government agencies. Tick-borne diseases in the U.S. Are a significant public health problem, and this is looking at the numbers of reported cases in the most recent year for which we have national surveillance data that's been submitted to us at CDC. And as you can see, Lyme disease is the most common reported tick-borne illness with over 33,000 cases, followed by spotted fever rickettsiosis with about 1/10 that many, followed by anaplasmosis, then babesiosis or ehrlichiosis, and then going down to tularemia, and then finally Poisson virus infection. These diseases not only are large numbers, but they've also been increasing over the last 20 years, basically, and this graph shows the trends of what we've seen for the 4 major reportable diseases. You can see that they've all been increasing. And some of the presentations we'll hear later on today will be talking a little bit more about these trends. Our webinar objectives are the following. The objective of our webinar today is to highlight the trends in emerging tick-borne diseases, including the geographic spread and discovery of new disease agents, and to bring attention to a novel and emerging non-infectious condition that's associated with tick bites in the United States. This is our topics and speakers. The first talk will be on the geographic expansion of Lyme disease and its vectors by Dr. Becky Eisen and Kirsten Kugler, who are here at CDC. The next talk will be on Borrelia mayonnae, a new cause of Lyme disease, and this will be by Dr. Ellie Thiel of the Mayo Clinic in Rochester, Minnesota. The next presentation will be an update on Borrelia miyamotoi, and this will be by Dr. Alan Barber at the University of California in Irvine, followed by an update on bourbon and heartland viruses by Dr. Nick Komar, also here at CDC. And then our last talk will be on meat allergy and Lone Star tick bites, and this will be by Dr. Scott Cummins of the University of North Carolina. So the way we'll handle questions and answers today is that we encourage you to submit your questions online through the webinar interface. Please also provide your identity and indicate the speaker to whom you would like your question to be addressed. And then the questions will be held until all the speakers have finished, and then those speakers will be able to address the questions that that are presented. So with that, only one last slide that I'd like to call attention to, and this is just for more information. This is where you can go to find out more about these tick-borne diseases. Also, as the operator mentioned, this webinar is being recorded, and it will be posted on our website for public viewing once it's finalized. There's a transcript, and all these things have been accomplished. So it will be several months later, but the other webinars are already there from previous years. So with that, I think we'll go to our first presentation, and this will be a joint presentation by Dr. Becky Eisen, who's a research biologist, and Dr. Kirsten Kugler, who's an epidemiologist. Both are in the Bacterial Diseases branch here in Fort Collins in the Division of Vector-Borne Diseases. And since joining CDC about 10 years ago, both of these speakers have been involved in domestic projects related to Lyme disease, as well as plague and tularemia and tick-borne relapsing fever. Both have also been very instrumental in international programs focused on plague surveillance and response. Dr. Eisen's research focuses primarily on medical entomology and vector-borne disease ecology. Dr. Kugler's efforts are focused on surveillance practice and better understanding risk factors for bacterial vector-borne infections. So with that, I will turn the presentation over to Dr. Becky Eisen.
Thank you, Ben. To get started, I'd like to review some of the basics of Lyme disease biology. Lyme disease is caused by the spirochetal bacterium Borrelia burgdorferi. In the United States, approximately 95% of all reported cases occur in the northeastern and north central states, but there are discrete areas of risk also in the Pacific Coast states. Borrelia burgdorferi spreads through the bite of infected ticks. In the United States, there are two major vectors, the black-legged tick, Ixodes scapularis, and the western black-legged tick, Ixodes pacificus. Each of these ticks have 4 life stages. Their generalized life cycle is shown to the right. Most humans are infected with Borrelia burgdorferi through the bites of nymphal ticks, although adult ticks are also capable of transmission. In most cases, ticks must remain attached to their host for at least 36 hours for transmission to occur. The nymphs are particularly dangerous because they're very small and can go unnoticed during this critical period. Supporting the notion that nymphs are the primary vectors to humans, the peak of Lyme disease occurrence corresponds with the timing of nymphal feeding in the spring and summer months. This map shows the generalized distribution of Ixodes pacificus in the west and Ixodes scapularis in the east. In this talk, I'll present data documenting the expansion of the reported distribution of these vectors over the last 2 decades. The expansion of the vectors range likely explains some of the observed increase in counties classified as high incidence for Lyme disease, which Dr. Kugler will cover in later slides. In the mid-1990s, David Dennis and others from CDC published a reported distribution map for Ixodes scapularis and Ixodes pacificus in the United States. Tick surveillance is not systematically or routinely conducted in the U.S. Therefore, to develop their distribution map, the authors relied on published records of the tick's occurrence at the county spatial scale. In addition, they sent questionnaires requesting information on tick collections to public health officials, acarologists, and Lyme disease investigators throughout the United States. What you're seeing in this map is the compilation of data derived from literature review and surveys. Counties considered to have established populations of Ixodes scapularis or pacificus are shown in red or green, respectively. Established populations were classified as at least 6 individuals or 1 or more life stages of the ticks recorded in a single year. Counties where the counts were not reported or failed to meet these thresholds were considered to have reported populations, and these are shown in yellow for pacificus and blue for scapularis. It's worth noting that the counties that are shown in white cannot be considered to be free of vector ticks. It's possible that the ticks may have been present, but not documented in literature or captured by the survey. We recently updated the tick distribution maps using similar methods to what I just described. From 1996 to 2015, we observed a 45% increase in the number of counties that have recorded the presence of these ticks. These vectors have now been identified in a total of 1,531 counties spread across 43 states. That is, Lyme disease vectors are present in at least 49% of counties in the contiguous United States. Ixodes scapularis now is documented to be established in 842 counties distributed across 35 states. This more than doubles the number of counties in which the tick is classified as established since the previous survey. The data presented here suggests that Ixodes scapularis over the past 2 decades has expanded from its northeastern and north central foci in all directions, with notable barriers including lakes and oceans. The 2 previously distinct foci in the northeast and north central states appear to be merging in the Ohio River Valley to form a contiguous single focus. These changes highlight the need for periodic update of the vector distribution maps. Although the presence of the vector is the first step in assessing the risk for Lyme disease, distribution data alone do not correlate well with the patterns of Lyme disease case occurrence. This can be seen by comparing the species distribution map on the far left of this slide with the distribution of reported cases on the far right. Notably, Ixodes scapularis is present in the southern states, but Lyme disease cases are rarely reported from the south. This apparent disconnect can be explained in part by differences in host-seeking behavior of northern and southern ticks and by infection rates in the ticks. In general, Ixodes scapularis from the north compared with the south are more likely to host-seek in a way that puts them into contact with people. In addition, infection rates are typically higher in ticks from the north compared with the south. The map in the center more closely resembles the map of reported Lyme disease cases. The observed density of infected host-seeking nymphs is shown as circles, and the model distribution is shown as a color ramp, where the warmer colors indicate a greater density of infected nymphs. Notably, the density of infected nymphs map shown here is the most recent of its kind, but it's based on the tick distribution from nearly a decade ago. The previous slide highlighted expansion vector tick populations. The distribution of human Lyme disease has also changed over a similar time frame. At this time, I'll turn the presentation over to Kirsten Kugler, who will describe the expansion of human illness in the United States.
Thank you. As you look at these two maps of reported Lyme disease cases, one from 2001 and one from 2014, you can clearly see the expansion over time of the geographic area in the northeastern United States where Lyme disease is occurring with frequency. While we can visualize this expansion through simple side-by-side map comparisons such as this, we wanted to quantify this geographic spread, and that's what I'm going to detail on the next few slides. For this effort, we first developed the methods and criteria for defining a high-incidence county based on reported Lyme disease cases. This methodology used SAT-scan software and compared the observed case counts in each county to the expected case counts. Expected case counts were generated given uniform risk across the U.S. while accounting for population differences. We then applied a cutoff to this observed to expected case count ratio in order to qualify a county as having high incidence. For this, we chose a ratio of 2. Therefore, for this analysis, high incidence counties were those where the observed number of cases was at least double the number that were expected. As we had to apply a cutoff somewhere, this cutoff of 2 naturally does not capture all counties with Lyme disease risk. We then applied these methods to data from successive 5-year time periods during the 20 years from 1993 to 2012. We tabulated the number of high-incidence counties in each of these 5-year time periods. Through this analysis, we demonstrated a roughly 300% increase in the number of high-incidence counties during this 20-year period, as well as geographic spread in both major foci. This table shows the number of counties that met the designated high-incidence threshold in each 5-year time period. Overall, you can see the number of counties increase from 69 in the 1993 to 1997 time period to 260 in the 2008 to 2012 time period, an increase of 277%. The increase was similar when data were separated by geographic region. The number of counties increased over 300% in the northeastern U.S. and about 250% in the north central U.S. The geographic centroids of these high-incidence areas did not shift substantially over time, indicating that spread occurred in all accessible directions rather than directionally, such as a predominantly northward or westward spread. This map provides a visual depiction of the geographic expansion in high-incidence counties. The time period when a county first reached the high-incidence threshold is indicated. The lightest color indicates the counties that reached the designated high-incidence threshold in the first time period, 1993 to 1997, whereas the darkest color indicates the counties that reached that threshold during the most recent time period analyzed. Here, you can see the spread over time of high-incidence counties outward in all directions in both major foci. And now, to summarize, through surveys and literature reviews, we've demonstrated that the number of counties with records of Ixodes scapularis or Ixodes pacificus has increased by about 45% from the mid-1990s to present. Separately, we've also demonstrated that the number of counties with a high incidence of human Lyme disease has increased about 300% since the early 1990s. However, human risk of Lyme disease depends on more than just vector tick presence. Specifically, it also depends upon the abundance of these ticks, their feeding habits, the prevalence of infection, and necessarily the human behaviors that bring people into contact with ticks. Expansion of areas with high risk of Lyme disease is likely multifactorial, and also it's likely to continue. The ongoing expansion of vectors and human disease that we've demonstrated here underscores the urgent need for effective and acceptable prevention methods. Here we've listed some references relevant to this presentation for anyone who's interested. And now we'll turn things back over to Ben Beard.
Thanks both to Becky and to Kirsten for those presentations, and that was very informative. And we'll go now to our next speaker, and this will be Dr. Ellie Thiel of the Mayo Clinic, and Ellie's presentation will be on Borrelia mayonii, a new cause of Lyme disease.
Thank you, Ben. Thanks for the introduction. It's a pleasure to speak to all of you today about our identification of Borrelia mayonii. So just to note my disclosures. And so to begin, as many of you know, the Borrelia genus can broadly be divided into two major groups, the relapsing fever Borrelia and those Borrelia species associated with Lyme disease, which are all members of the Borrelia burgdorferi sensu lato complex. There's over 15 genospecies within this complex. However, 3 of them are most commonly associated with Lyme disease, including Borrelia burgdorferi sensu strep 2, which I'll just refer to as Borrelia burgdorferi, and that's really the main cause of Lyme disease in North America. And then we have Borrelia guerinii and Borrelia afzelii, which are geographically localized to Europe and Asia. So as was discussed, these spirochetes are transmitted through Ixodes species ticks, and in both Europe and North America, Lyme disease is considered to be the most common vector-borne illness with, as we just heard, over 33,000 cases reported in the U.S. in 2014. So diagnosis of Lyme disease relies on both clinical evaluation and laboratory testing, particularly in cases where classic signs such as erythema migrans rash are absent. And the 2 main types of laboratory tests that are available include serologic evaluation using the 2-tiered testing algorithm, which I'll discuss a little later on, and then also molecular testing by real-time PCR for detection of Borrelia DNA. PCR tests are typically positive during the acute disease and prior to development of an antibody response. However, this methodology has a fairly low clinical sensitivity, with blood being positive in only about 50% of patients with a classic erythema migrans rash. So PCR for Borrelia DNA is really considered to be an adjunct test at best for Lyme disease. Briefly, however, our Borrelia PCR assay at Mayo Clinic, it's a FRET-based assay which uses hybridization probes targeting the plasminogen binding protein, or the OPA1 gene, that's shared amongst the Borrelia burgdorferi complex members. And using our assay, we can actually differentiate Borrelia burgdorferi from Borrelia guerinii and Borrelia afzelii by melting temperature analysis, which very briefly just relies on probe detection of species-specific pleomorphisms in the target OPA1 gene. So to show you what this looks like, this is a screenshot of our PCR melt curves, and as you can see, we get different probe melting temperatures for the different species as shown on the x-axis. So for Borrelia burgdorferi, the probe melt temp is approximately 65 degrees, whereas for the European species, the melt temp is roughly 55 degrees. So with that background, in June of 2013, we noticed this distinct but pretty atypical peak that was right between our validated melt temps for Borrelia burgdorferi and the European species. So a number of things happened all at once, but one of the first questions we asked was, has this atypical peak appeared before? And to answer this, we retrospectively reviewed our data since 2003, which is when we first brought this assay on, all the way through 2014. So between those 11 years, we had performed Lyme PCR on over 100,000 specimens from patients in all 50 states. And interestingly, we did not detect a similar atypical peak in any of the samples tested between 2003 and 2011. So we So we didn't detect an atypical peak in over 66,000 specimens tested between those time frames. However, between 2012 and 2014, we detected this atypical peak in 6 samples out of over 33,000 specimens tested. And interestingly, these were 5 whole blood samples and 1 synovial fluid that were all from residents of Minnesota, Wisconsin, and North Dakota. So the next question became, well, is this possibly a novel or a new Borrelia species? And so to investigate this, we knew we had to perform some sequencing studies, and thankfully we save all of our Lyme PCR positive samples indefinitely. And of the 6 positive samples, we still had 4 in storage with sufficient DNA to be able to perform multi-locus sequence analysis for a number of genes, including 8 concatenated housekeeping genes that were previously used to define Borrelia burgdorferi sensu lato complex members. So what I'm showing you here is a phylogenetic tree based on our sequence analysis, and what you'll notice is that for the 4 available patient samples, they all clustered together, as indicated by the red box, and they were significantly removed from Borrelia burgdorferi, Borrelia pfellii, and Borrelia guerinii, as indicated by the green boxes. So notably, the highest similarity of these samples with atypical peaks was to Borrelia burgdorferi at approximately 95% sequence homology. And in order to be considered a unique species within this complex, sequence homology must be less than 98.3%, which it is, confirming that we're dealing with a novel Borrelia genospecies, and we've proposed to name it Borrelia mayonii in honor of the Mayo brothers. So while working on our sequencing studies, our colleagues at the CDC were able to grow and culture the spirochete from 2 different whole blood samples, one of which was collected 1 day post-illness onset, and you can see a representative image of that spirochete in the picture shown here. Also, through some additional quantitative PCR studies, we were able to determine that, in general, the bacterial load in the 5 whole blood samples we had ranged from approximately 10 to the 5th to 10 to the 6th genomes per mil, which on average is 180-fold higher than the bacterial loads from patients infected with Borrelia burgdorferi. So we see a much higher spirochetemia in patients with Borrelia mayonii compared to patients with Borrelia burgdorferi. So what about the clinical features of these patients? Well, as I mentioned, all 6 of them were residents of either Minnesota, North Dakota, or Wisconsin. And while 2 of them had known tick bites, all had tick exposures in high-risk regions of Minnesota or Wisconsin, as I'll show you on a subsequent slide. 4 of the 6 patients were male, and their ages ranged from 10 to 67 years. 5 patients presented with a fever and headache, and 3 of them had potential neurologic involvement, including profound somnolence, confused speech, and blurry or double vision. 4 patients had a rash, but only 1 of those reported an erythema migrans-like lesion. The rest had a diffuse macular papular type rash, as shown in the image here. This is our 10-year-old patient. You can see that the rash is on the trunk and the extremities, but also this individual reported it on the palms and soles of his feet as well, which is interesting. And then importantly, 5 out of 6 patients all recovered completely with antibiotic therapy, though the individual who presented with arthralgia did continue to have residual joint pain. Okay, so we were able to detect Borrelia mayonii with our PCR assay. However, the current recommended diagnostic testing methodology for Lyme disease is through serologic detection of antibodies using the 2-tiered serologic testing algorithm. So as a very brief review, you'll remember that this algorithm begins with an initial screen, which is most often done by an EIA. For screen-negative individuals, an alternative diagnosis should be considered, or if the sample is collected too early following symptom onset, a convalescent sample should be submitted. And then for screen-positive or equivocal samples, supplemental testing by Western blot specific for IgM and or IgG antibodies, depending on the duration of symptoms, should be performed. So this is a summary table of the serologic results that we have for 5 of the 6 patients. And starting with patient 1, we had a single serum sample for this individual collected 6 days post-symptom onset. And you can see that the C6 screening ELISA and the IgM were both positive, and then the IgG blot was not surprisingly negative. For patient number 3, we had 3 serum samples collected on days 2, 29, and 104 post-illness onset. And you can see that the C6 seroconverted from equivocal to positive by day 29, at which point IgM was also positive. Interestingly, though, even at day 104, the IgG blot remained qualitatively negative. Patient number 4 had a single available serum sample collected 266 days post-onset, and while both the C6 and the IgG blot were positive, the IgG only had 5 bands present, which you'll remember is the minimum band number for positivity that's required. Patient 5 was C6 positive on both day 3 and 32 post-illness onset, and he did seroconvert to IgM blot positive. And then finally, patient 6 was seronegative, which is not surprising since his serum sample was collected very early on in his disease course. So collectively then, this data indicates that the C6 ELISA and possibly other VLSE-based assays will detect antibodies to Borrelia mayonii. And while the current IgM blots appear to perform well for Borrelia mayonii, it seems that the IgG blots are not as sensitive. However, with an N of only 5 patients, we really need to continue to evaluate this and plan and hope to do so this coming tick season. And then finally, I wanted to conclude with a couple of words on what we think the vector for Borrelia mayonii is. We decided to focus on Ixodes species ticks, and we did a number of tick drags near one of the possible patient exposure sites in Wisconsin that's indicated by the arrow here. And then we tested these ticks in a number of archived ticks from previous tick drags by our PCR assay for Borrelia mayonii. And so what you can see is that we detected Borrelia mayonii in 19 out of the 658 ticks we had, so that's about a positivity rate of roughly 2.9%. And for comparison, we detected Borrelia burgdorferi in almost 30% of them. We also sequenced the DNA that we got from these ticks and confirmed that it was the same organism we were detecting in our patients, I should say. So it appears that Ixodes species ticks are a likely vector for Borrelia mayonii as well. So to conclude, we've identified a novel member of the Borrelia burgdorferi sensu lato complex, and we've proposed the name Borrelia mayonii for it. From the 6 patients we've studied to date, Borrelia mayonii appears to cause similar but more severe disease compared to Lyme disease caused by Borrelia burgdorferi. Notably, we found Borrelia mayonii predominantly in whole blood, which is a specimen source that's rarely positive for Borrelia burgdorferi. We've also shown that Borrelia mayonii infection is associated with a higher bacterial load, about 180-fold higher than that observed for Borrelia burgdorferi. And then finally, we've shown that Ixodes scapularis ticks are a possible vector for this new species. And then finally, it's really important to acknowledge that this whole discovery and project would not have been possible without the close collaboration of the Minnesota, Wisconsin, and North Dakota state health departments, as well as the CDC and the University of Wisconsin. And so with that, I will thank you for your attention and hand it back over to Ben.
Thank you, Ellie. And I have to apologize that I actually failed to properly introduce Dr. Thiel. And so I'll say at this point that she is an assistant professor of laboratory medicine and pathology and also the director of the infectious diseases serology lab at Mayo Clinic in Rochester, Minnesota. She received her PhD from the University of Wisconsin in Madison in medical microbiology and immunology, and she completed her clinical microbiology fellowship at Mayo Clinic. Ellie's certified by the American Board of Medical Microbiology, and Dr. Thiel's research interests include developing and evaluating improved methods for detection of vector-borne infections. So thank you for that informative presentation, and sorry that I introduced you after the fact, but maybe we'll get back on track here. And so I'll go ahead and introduce our next speaker. and this will be Dr. Alan Barber. And Alan's a professor of medicine and microbiology at the University of California, Irvine School of Medicine. He's a co-discoverer of the cause of Lyme disease and a leading Lyme disease researcher. For more than 30 years, Dr. Barber has been studying the pathogenesis of Borrelia infections, the most important of which are relapsing fever, one of the neglected tropical diseases, and of course Lyme disease, which is the most common arthropod-borne disease in North America and in Europe. For much of that time, his emphasis has been on understanding the genetics, disease mechanisms of pathogens, and also the immune responses of the host of the microbes. So Alan's been a leader in the field for many years, and so I want to thank you for joining us, and we'll turn the presentation over to Dr. Barber now.
Thank you very much, Ben. That gives a little bit of background. The first slide shows that, like a lot of things, everything isn't new under the sun. More than 100 years ago, Dr. Tyler, one of the pioneers in parasitology, described an organism they found in ungulates in Africa, and you notice this was reported by Dr. David Bruce, for whom Brucella is named. So some of the early microbiologists noticed this organism, and then it kind of slipped below the radar. But some of the features of this, which came to be called Borrelia tyleri, are that it caused this bacteremia in cattle. It moved around the world with the movement of cattle, and it was carried by hard ticks instead of soft ticks. People knew about soft tick-borne relapsing fever, but this was something different. And they also noticed that it had transovarial transmission, and to date, no one has been able to cultivate this organism. And then something like this popped up again in the 1980s, soon after Borrelia burgdorferi and the other organisms that have been mentioned, Ixodes and Green, were discovered, and there were conferences about this. But one of the early conferences on Lyme disease in Vienna, there was this report by Dr. Crumpets described this organism that was behaved very differently from what we understood Borrelia burgdorferi did. It achieved high levels in experimental animals, and it seemed to be associated with the Ixodes ricinus ticks. But nothing much was done with that thereafter. It was difficult to cultivate it, and so, again, it slipped below the radar again. Until this report from Japan in 1995, in which Dr. Fukunaga, one of the early researchers in Lyme disease in Japan, and his colleagues described this new organism that they found in Ixodes persulcatus ticks, which were known to transmit Lyme disease in Japan and throughout Asia and going into Russia. This other organism, which they named Borrelia miyamotoi after Professor Miyamoto, who had been a long and distinguished career in entomology in Japan. And it fell not like the Lyme disease group, but within the relapsing fever group. But it was not, thereafter, it was not really associated with any infections. It was interesting curiosity. Some of us began to study it, but we wasn't sure what the public health significance of this might be. But it was discovered also in North America. This is some work coming out of Gerlin Fish's lab, led by Glenn Scholes, who found this organism in larval ticks, Ixodes scapularis ticks. And this would have been, against the what we would expect for Borrelia burgdorferi, which was not known to be transmitted transovarially. So to find a Borrelia in larval ticks was unusual. They did some very nice studies in working this up, found it was transovarially transmitted, and that it also fell within the relapsing fever group of species. Subsequently, it was found in Ixodes ricinus ticks, in Ixodes persulcatus ticks in Russia, Ixodes pacificus ticks in California, and also in the north central United States. So this shows kind of the relationship between these group of organisms that are distinguished from the Lyme disease group. We know that Borrelia miyamotoi is carried by Ixodes ticks of at least 4 different types, maybe more. Another tick, Borrelia lonsdari, another Borrelia is carried by amblyomma ticks, and there are other related Borrelia by other amblyomma ticks in North America. We see the Borrelia tyleri there associated with rhipicephalus ticks, and there's and below that in the soft ticks, our gassed ticks are the relapsing fever species that we've known about for many years, and also an agent of bird disease, Borrelia anserina. So now we can see that this organism is carried by the same ticks as what carries Lyme disease throughout the temperate regions of the northern hemisphere. And looking at this a little more closely, we can see at the top of this slide the sort of the phylogeny of some of these ticks, the hard tick-associated relapsing fever group. Up near the top are the miyamotoi from California, from Connecticut, Japan, Germany, compared to an isolate of Borrelia lonsdari from Texas. And then below that, the group of the soft tick-associated relapsing fever species, including the one from Africa. Now, what we and others have noticed is that with any of these tick associations, such as with Ixodes ricinus in Germany, persulcatus in Japan, pacificus or scapularis, but within each of these associations, all the isolates or the PCR amplifications that we and others have looked at seem to be identical or near identical. And this would be different from what has been noticed for Borrelia burgdorferi, which has a sort of a complex string structure, and anywhere it's found, there may be up to 10 to 15 different strains in any one area, even a small area. So miyamoto seems to be different and much more associated with the tick vector, the specificity there. But it is distributed across much of the same area that Borrelia burgdorferi is found in North America and also extending up into Canada. And roughly the ratio of Borrelia burgdorferi to miyamotoi and the nymphal ticks, Ixodes, scapularis, or pacificus ticks, collected in these areas is about 10 to 1. It can vary a little bit about that, but it's a remarkable fairly constant relationship between that, except in California, and for all we know in other parts of the far west, where Ixodes pacificus occurs, and there the ratio is about 1 to 1, and in some places of California, miyamotoi prevalence in nymphal ticks exceeds that of Borrelia burgdorferi. So that was what was known. A lot was known. This was distributed around the world, within North America, but still there was no association with disease until this report from Russia, which involved a close collaboration with Derlin Fish and Peter Krause at Yale of cases what appeared to be relapsing fever, something close to that in Russia. And this paper describes this in detail and the location of where the cases occurred, and the ones that were illustrated had a relapsing fever type of fever course. If you just showed this slide without knowing anything more about it, you would think that this might be a relapsing fever. But the majority of the patients did not have a relapsing course, perhaps because they were treated early or for whatever reason, or it just doesn't happen very often. But it does indicate that relapsing fever type of picture can occur with these cases. And the map down in the left-hand corner shows the distribution of that and approximately overall about 2% of the ticks, ricinus ticks or persulcatus ticks in this area. So then, with that evidence, Peter Krause and his colleagues at Yale and our contributions to this began a seroprevalence study, and there was 2 of them. One reported in the New England Journal, and the next, a larger study reported in Emerging Infectious Diseases. And basically, the background rate of seropositivity, which may be false positives, with this GLP-Q assay. The GLP-Q antigen is a antigen discovered by Tom Schwann and his colleagues that relapsing fever organisms make, but Borrelia burgdorferi and the other Lyme disease groups do not make and do not even have the gene. So it's specific for the relapsing fever group, including miyamotoi. And so this has worked well with relapsing fever diagnosis, and also now works well as really the sole serology for doing a miyamotoi disease at this time. So as I said, the background rate seems to be about 1%. Whether this is false positive or just individuals who had been infected in the past is not clear. But in high-risk areas where there's, like on islands off the coast which have a lot of Lyme disease, about 10% of the individuals are seropositive for Borrelia burgdorferi and about 4% for miyamotoi. And it seems to be occurring also in individuals who have a viral-like illness during the summer. Certainly not all of them, but some of them. And another study that came out of a laboratory in Massachusetts, a laboratory that received blood from all over the Northeast and tested for different tick-borne diseases, usually during the tick transmission season. And this was reported by Malloy in Annals of Internal Medicine. And overall, from their bloods that they received, about 1% were PCR positive for miyamotoi, and they confirmed this by sequencing. And of the people that they could get the common histories from, most had an undifferentiated viral-like illness with fever, and about a quarter of them were hospitalized because of suspected sepsis. They often complained of a severe headache. And at the time when the bloods were drawn, and going back retrospectively, only 16% of these people had antibodies to GLP-Q at that time. So this is the top distribution of cases is from Malloy, and below is a fairly typical distribution of cases for Borrelia burgdorferi. And you can see there's a different time of the peak of miyamotoi cases. And this is not clear what it's from, but it's possibly because of transmission by larvae. So miyamotoi may be transmitted by larvae. And the other factor we need to consider is that because the organism is already in the salivary glands of the tick at the time of biting, the transmission may occur very quickly, and so the window of 36 to 48 hours might not apply for miyamotoi. So people may get it soon after tick bite starts. Now I mentioned the disease can be more serious. There are 2 cases of meningoencephalitis, one in the United States and the Northeast reported here, in which they could actually see the organism in the CSF. There's been another similar case from the Netherlands. Both of them were older patients who had an underlying immunodeficiency, but their symptoms went on for weeks to months, so it can potentially cause a chronic disease of the central nervous system that may go unnoticed or not be obvious. And it's well known that the relapsing fever species have a tropism for the central nervous system. So some of the key points of difference from Lyme disease is have a higher temperature. They may have a relapse of fever, a viral-like illness, the headache. There may be co-infection with Borrelia burgdorferi, and you may have erythema migrans, a rash, but it's not noted to be seen in just pure miyamotoi infection. More likely to have hospitalization, with miyamotoi, more on the lines of anaplasmosis. There may be a low platelet count, abnormal liver enzymes. So again, the most difficult differential diagnosis is with human granulocytic anaplasmosis. Well, isolation is not easy. Our recommendations are, if there's suspected cases, to inoculate immunodeficient mouse with blood or CSF to isolate the organism. There are some published modifications to BSK that have reported to have success in growing them, but these have not been easily reproduced between laboratories. I suspect it's the source of the BSA is important, and overall the yield is much lower than with burgdorferi. There are other more involved ways of doing it to isolate this organism, involving taking female ticks and looking at the offspring of the positive ones and putting them onto mice. The diagnosis, we can go back and to do some of the old techniques like phase or dark field microscopy. It might be noted on that. The white or thin smear, but the organism may not achieve very high levels to see it that way, so I recommend doing a thick blood smear with fluorescent antibody to Borrelia. PCR, this is commercially available and also available in other laboratories with specific probes for this. And then the serology is the GLP-Q. There may be a false positive of Lyme disease ELISA with whole cells because of cross-reactivity, but the Western blot would be negative. The treatment would be similar as for Lyme disease and relapsing fever, which means a tetracycline or beta-lactam. So a doxycycline, I think, would be the first choice because it would be very difficult to rule out anaplasmosis at the time of presentation, and then intravenous beta-lactams for CNS disease. But we don't really know the susceptibilities of these organisms. It's all empirical studies so far. So just to finish up, the working name of the disease is, we suggest hard tick-borne relapsing fever to differentiate it from the other forms. It's not as severe, as distinctive as other forms of relapsing fever, but we don't know about the consequences, about equal risk for getting it in California, it seems. The high specificity of tick vector means a different kind of ecology than what we've seen with burgdorferi, and it's possible that there could be blood acquisition. This is a bibliography that will be available with the transcript, but I can make it available through my Twitter feed at Alan Barber, lowercase, all one word. And there's also a YouTube video that you could find that shows this in the blood of plasma. And I thank the other people involved with this.
Thanks, Alan. We appreciate that presentation. And so now we will move on to our next speaker. And this speaker, I'm sure I get the right presentation loaded here. Our next speaker will be Dr. Nick Kumar, and Nick is a vertebrate ecologist in the Arboviral Diseases branch here at CDC in the Division of Vector-Borne Diseases in Fort Collins. Nick's research interests are transmission ecology of zoonotic arboviruses and environmental surveillance for arboviruses. He has focused most of his efforts on tracking and understanding the ecology of West Nile virus through the Americas. And Nick has published more than 120 peer-reviewed scientific journal articles on arboviruses. So with that, I will turn the mic over to Nick.
Thank you, Ben. Today I will be reviewing the important tick-borne viruses that are known to cause disease in the USA. In general, tick-borne viruses are rare in the USA. They are more common in Northern and Eastern Europe and Northern Asia, where tick-borne encephalitis is a significant public health issue. Southern Asia has suffered recent outbreaks from chiastin or forest disease caused by a tick-borne flavivirus. Within the last decade, another area where a major tick-borne virus has emerged is East Asia, where a novel tick-borne flebovirus has caused thousands of cases of severe fever and thrombocytopenia syndrome. But fortunately, in the U.S., tick-borne viruses rarely cause human illness. However, the Earth is in a state of rapid ecologic change, and the incidence of tick-borne viral diseases may be changing. It is important that we stay vigilant. In order to recognize a new disease emergence, we must be familiar with the viruses that are already among us, and that is the objective of this lecture, to review what we know about the tick-borne viruses already causing illness in the USA. There are just 4 viruses I will be reviewing. These are Colorado tick fever virus, Powassan virus, Heartland virus, and Bourbon virus. They are each very different from each other. They pertain to 4 different virus genera listed here as Coltivirus, Flavivirus, Phlebivirus, and Ixodes, each from its own distinct virus family. Listed here as Reoviridae, Flaviviridae, Bunyaviridae, and Orthomyxoviridae. Each of these viral families has a different molecular biology and thus a distinct evolutionary history. As you can see from the last column on this slide, none of these viruses cause more than a mean of 10 recognized cases per year nationwide. Therefore, being rare causes of disease, they don't receive much attention from the public. Most physicians have never seen a case of one of these exotic diseases, and therefore general awareness of these viruses among the medical community is low. Needless to say, the public health burden of these or similar viruses in the USA is probably much larger than recognized, and even an outbreak could be overlooked. We'll start with Colorado tick fever virus, as it is the cause of the highest number of human cases of tick-borne virus disease in the USA. Colorado tick fever virus persists in nature in an enzootic cycle that involves the Rocky Mountain wood tick, Dermacentor andersoni, as the vector, and a variety of wild rodents, such as golden-mantled ground squirrels shown here, that serve as vertebrate reservoirs. Other rodents that serve as vertebrate reservoirs for this virus would include other ground squirrels, tree squirrels, field mice, voles, and chipmunks. Human beings get infected incidentally when an infected tick accidentally bites a human being. Human beings are not considered to be competent hosts, which means they do not become infectious to ticks. The enzootic cycle is limited geographically by the range of the tick vector, shown on this map as light blue. Dark blue indicates the county of residence of people which became ill with the virus for a series of 83 human cases reported to Arbonet, which is our national surveillance system for arthropod-borne viruses in the USA. All cases were acquired in western states where local transmission of Colorado tick fever virus has been reported. Since this map was made, the incidence of reported cases has remained stable at less than 10 cases per year. The disease caused by Colorado tick fever virus is mild and non-life-threatening, although occasional cases of hemorrhage, encephalitis, and even death have been reported. Certain common symptoms are fever, chills, headache, body aches, and fatigue. Some patients develop a skin rash, sore throat, or abdominal pain. Patients typically become ill after a vacation in a natural setting where they encounter plenty of wildlife. Symptoms resolve without treatment. Powassan virus is the cause of the second highest number of human cases of tick-borne virus disease in the USA. Powassan, however, causes a more severe disease compared with Colorado tick fever virus, including encephalitis, which is inflammation of the brain. It is a close relative of the tick-borne encephalitis viruses in Eurasia. These are all flaviviruses, which include mosquito-borne encephalitis viruses like Japanese encephalitis virus and West Nile virus. Powassan virus occurs in a natural transmission cycle between small mammals and ticks in the genus Ixodes. There are two main genotypes of Powassan virus. Lineage 1 Powassan virus is transmitted by Ixodes cooki, the woodchuck tick. Lineage 2 Powassan virus, also known as deer tick virus, is transmitted by Ixodes scapularis, the deer tick, also known as the black-legged tick. Immature Ixodes ticks become infected after feeding on one of the vertebrate reservoirs of Powassan virus. For example, a white-footed mouse, shown here, can infect a deer tick, or a squirrel or a woodchuck can infect a woodchuck tick. Months later, when these ticks are ready for their next blood meal, they could transmit the virus incidentally to humans, which are considered to be dead-end hosts. This means that humans are not an important part of the virus's transmission cycle and are not likely to be infectious to another tick. The disease caused by Powassan virus is typical of other encephalitis viruses: fever, headache, vomiting, and neurologic symptoms like weakness, confusion, loss of coordination, difficulty with speech, and seizures. There is no antiviral treatment, but with good supportive care during hospitalization, survival of severe cases should be about 90%. But persistent neurologic sequelae are common. This is a very nasty disease. There are two geographic foci in the United States, which reflects the two areas where Ixodid ticks are increasing in prevalence: the upper Midwest and the Northeast. Minnesota, New York, and Wisconsin have suffered the brunt of these cases in recent years. This bar graph shows what appears to be a trend of increasing incidence. When the case numbers are this low, it is difficult to understand the true cause of such an apparent trend. Improvements in surveillance may be a contributing factor. Fortunately, the numbers of cases have not continued to rise since 2013. However, this is a situation that merits continued vigilance. Okay, the third most important tick-borne virus disease in the USA is heartland virus. It was only recently discovered in 2009 when it was isolated from a human patient at the Heartland Regional Medical Center in Missouri. We are still learning about this virus, but we believe it is transmitted incidentally to people by the Lone Star tick, Amblyomma americanum. We know that several mammal species are infected at high rates, particularly white-tailed deer. In Missouri, in counties where human cases have occurred, about 50% of the deer sampled have antibodies, which indicates past infection. We do not know for sure yet, but deer may be the vertebrate reservoir. Raccoons also are frequently infected, and a high percentage circulate antibodies in their blood. People are not thought to play an important role in transmission and are probably dead-end hosts for heartland virus. The disease caused by heartland virus has only affected men, and it is unknown whether this is a biological risk factor or a behavioral risk factor. The men who have been infected spend lots of time outdoors in heavily tick-infested habitats. The disease syndrome is similar to other tick-borne illnesses like ehrlichiosis, and like other tick-borne viral diseases, does not respond to antiviral treatment. Cases of heartland virus are detected when the patient fails to respond to antibiotic treatment. Symptoms include fever, fatigue, headache, body aches, nausea, loss of appetite, and diarrhea. Signs include low white cell count and low platelet count. Hospitalization can last several weeks, and the case fatality rate is roughly 10%, although this is based on a very low sample size. Fewer than 20 cases have been reported to the CDC, mostly from Missouri, but also from Oklahoma and Tennessee, from areas where Amblyomma americanum ticks are plentiful. This map, produced by Becky Eisen's team here at CDC, shows the range of the Lone Star tick, which covers a much larger geographic area where human cases have not yet been reported. Considering the large range of the Lone Star tick, my group collaborated with state and federal wildlife agencies to obtain and analyze blood samples from wildlife likely to be exposed to Lone Star tick bite, mainly white-tailed deer, raccoon, and coyotes. On this map, the blue circles represent the locations where antibody-positive animals were detected. Thus, serologic evidence of infection in mammals was reported from 11 additional eastern states from Texas to New England. So this is further evidence that human cases may be occurring elsewhere but not yet reported. Finally, the last virus I will discuss is called Bourbon virus. It was recently isolated from the blood of a patient who died following a tick bite in Bourbon County, Kansas, eastern Kansas, in June of 2014. Lab studies at CDC have confirmed that this virus replicates well in tick cells and mammalian cells, suggesting that its natural transmission cycle may be similar to heartland virus. However, we still know very little about this virus at this time. The disease syndrome is very similar to that of heartland virus. Interestingly, this virus is a distant relative of influenza viruses, which pertain to the same viral family, Orthomyxoviridae. Other closely related pagodaviruses are known to cause tick-borne disease in Asia and Africa. One other pagodavirus is known from the USA, called Aransas Bay virus, but it has not been associated with any disease. Bourbon virus was discovered as part of an active surveillance research project studying heartland virus. It is a reminder that other tick-borne pathogens are probably circulating in the USA and can be easily overlooked. And with that, I can entertain questions later during the question and answer period. Thank you very much.
Thanks, Nick. Really appreciate that. And now we will go on to our last speaker for the webinar. And pull up the presentation here. And the speaker will be Dr. Scott Cummins, and Scott is an associate professor of medicine at the University of North Carolina in Chapel Hill. Dr. Cummins sees patients in the UNC Allergy Clinic and maintains an active research laboratory. His primary research is related to the role of tick bites in the development of alpha-gal allergy to better understand how and why the allergy response occurs so that patients with this and other food allergies can be effectively treated. And this will be the topic of his talk today. So with that, I'll turn it over to Dr. Cummins.
Thank you, Dr. Beard. These are my disclosures and some objectives for today. I want to make the point very early on that the work I'm going to show is that of a team, because it takes a village often to do science, and so none of this is done in isolation, and I want to be clear about that. Our story actually starts in the cancer literature. In the late 2000s, there was an awareness amongst oncologists, particularly those in the southeast who were using a medication called cetuximab, that there was an increased incidence of hypersensitivity reactions at first infusion of that monoclonal antibody. There was work then done in the lab of Dr. Platts-Mills to show that when cetuximab was given and produced through SP2O cells, which are a mouse myeloma cell line that actively glycosylate with a sugar called alpha-gal or galactose alpha-1,3 galactose, that there is sufficient IgE binding, which are the numbers shown here and circled in blue, to cetuximab as it is made and marketed. However, if cetuximab is placed in a cell line that does not use the same galactosyl-transferase shown in CHO cells in the second column, which lack the ability to put alpha-gal on cetuximab, the binding seen from the SP2O cell line diminishes. So actually, it's completely gone. So what this set of experiments told us was that in patients who were receiving cetuximab in the South and reacting very quickly on first infusion, that this appeared to be due to a pre-existing IgE antibody that bound specific to alpha-gal. And what is alpha-gal? It's a carbohydrate that's synthesized by the galactosyl-transferase enzyme written there. It is present on the tissues of all lower mammals and really may not be identified or be important for the story that my lab works on, except for the fact that that is true. So any patients who have IgE to alpha-gal and consume beef or pork or lamb are coming in contact and consuming the sugar to which they are allergic. And awareness of this led us in 2009 to publish a manuscript of 24 patient reports throughout Virginia and southern Missouri of patients who described delayed reactions occurring 3 to 6 hours after eating red meat, where they would have anaphylaxis or itching or hives or gastrointestinal distress. In these patients, their prick tests were often fairly small, and their intradermal tests, however, were fairly striking. An example of this is shown here, where a prick test on the left has a modest response at best. However, intradermal testing shows significant skin responses, indicating that these patients have an IgE antibody response that binds an antigen in beef, pork, lamb, and that would be true in these patients of any mammalian antigen. So if we looked in the blood work, for instance, of a patient who would be alpha-gal positive, and this is 24, 25 patients showing IgE to alpha-gal in the green circle. In the blue circles there, beef, pork, and lamb levels would also be positive. Typically the chicken, turkey, and fish are negative. Cat and dog as mammalian antigens are also positive. As long as cat and dog are not consumed, then this does not pose a threat for reactivity as best we can tell. The initial reports were due to patient recollection and really had not shown formal demonstration of the delay. So our goal in early 2010, 2011 was to begin to challenge patients with mammalian meat and try to document a significant delay, as this was relatively unprecedented in the world of food allergy. So this is the picture of a subject who consumed roughly 2 pork sausage patties. This is a pre-photo before we began the challenge, and he was actually released after 6 hours with a small itch and a very subtle hive on the small of his back. However, he called me from the road and wanted me to know that the itching on his back had become more prominent, and when he got home, in fact, his skin was much redder and quite itchy. Despite treatment, this had progressed to an additional spread of whelps throughout his his upper torso, he never had any difficulty breathing and was not in distress other than significant and severe itching. Other challenge photos that we did also show, as patients report to us, significant palmar erythema and swelling often associated with these reactions shown here, as well as significant urticaria or hives that can follow consumption of mammalian meat in these patients. At times, the reactions can be simply discrete areas of urticaria, shown here on the right flank of a patient eating pork sausage challenge, but also certain patients have very specific gastrointestinal symptoms without much in the way of cutaneous signs. So there is an individual component to this allergy as well. We were initially struck by trying to understand what could have created this risk, because most of these patients safely eaten beef or pork or lamb for their entire adult lives, and then suddenly begin to have difficulty with it, and often these symptoms occur at nighttime after a dinner meal of mammalian meat. And one of the intriguing possibilities was that ticks, in fact, could be creating a risk for this. So when we reviewed the geography of the cetuximab hypersensitivity reactions shown here in the red states, as published by Dr. O'Neill in 2007, we were struck both with the overlay of Rocky Mountain spotted fever incidents, the distribution of our cases that we had knowledge of in 2011, as well as the published range of the Lone Star tick. Now, certainly we were not indicating that Rocky Mountain spotted fever was transmitted by amblyoma, but merely that the geographical overlay of these maps was fairly striking as it related to our patient population. And so we undertook a telephone survey of subjects that we had enrolled and began to ask them whether they in fact reported tick or in this slide, chigger bites. I include chiggers not because I think that they are ticks, but because seed ticks are, I think, often mistaken for chiggers. And so to get a more accurate representation of a patient's full exposure history, we asked about tick or chigger bites. And you see in the red triangles that most of the patients reported a history of tick or chigger bites. In the black triangles, there are a few which we were unable to obtain history for, and there was a small group shown here that specifically denied tick bites, and it turns out that this group of patients probably has more severe atopic dermatitis or eczema than actual meat allergy in retrospect. So that began to make us, get us think that perhaps tick bites were a part of this story, and we've always been impressed by how many eggs can be laid by a single adult female ticks, and equally with the foot shown here, how many bites can happen if one passes by the wrong leaf, so to speak, in the woods. What I want to use this slide also to make the point that in this subject who had a negative alpha-gal test in May of 2007 and then suffered numerous seed tick bites in August of that same year, shown here, their follow-up alpha-gal level in October was 48, and then rose again to 130 in November. Meanwhile, the levels for chicken, turkey, cockroach, or dust mite all remained negative. So this was a very specific response for alpha-gal in this patient, and began to give us a little more evidence early on that perhaps ticks were important. We've seen this a few times now where we follow patients or subjects longitudinally. Evidence here from a single patient who was diagnosed in early July and then had 50 or so bites from larval ticks over Labor Day weekend last year, shown here with the red bar. And you see a nice rise in the alpha-gal specific IgE, which is a marker we use of the meat allergy. And then it does appear that this begins to decline over time. And we've seen this in other subjects that we've had the ability to follow or for a longer period of time, where their alpha-gal level, again shown in orange, appears to spike after tick bites, shown here with the red lines, and then again appears to naturally resolve over time. So in summary, hopefully I have taken a few minutes here to suggest that tick bites can affect the alpha-gal specific response in some patients, and that we believe this sensitization appears to wane over time, that additional tick bites appear to lead to more significant increases. And I didn't show any data related to this today. There is at least an initial report that's been published linking alpha-gal present in the GI tract of Ixodes ricinus in southern Sweden. So that's all I have for today, and I'll be happy to turn the floor back over to Dr. Beard.
Thanks, Dr. Cummins. We appreciate that presentation. And so now we will open the webinar up to public questions for any of our speakers. And as I mentioned before, if you can identify yourself and also mention which or specify what speaker you'd like the question to be addressed to, if you would like. So we can take questions now. I'll give it just a couple of minutes longer here, and if not, we will bring the webinar to a close. So there's one question here for Nick Komar. It's a comment and a question calling attention to the fact that there are products available that keep ticks off of hunting dogs, horses, and deer, and that they seem to be pretty effective. And this is coming from Bourbon County. And the question is, is there interest in seeing the test results? So whoever the speaker is, he'll have to send that directly to Dr. Kumar by email if he wants, Dr. Kumar, if he wants that question answered.
Yeah, I look forward to seeing the email. Thank you. Do they have my email address?
If you want to give it, that would be good.
Okay, so it's nkomar, my first initial and then my last name, nkomar @cdc.gov. Thank you.
Okay, thanks. There's a question here, and it is, in light of the increases in the spread of ticks and Lyme disease, how can people prevent tick bites? And maybe I will channel that question, first of all, to Drs. Eisen and Kugler, and then to anyone else who would like to take a shot at that.
Yes, thank you. Thank you, Ben. You know, we don't have that many great options for prevention of tick bites that have been shown to greatly reduce incidence of human disease. But nevertheless, there's a lot of things that people can do to minimize their own risk of getting tick bites, and that includes using insect repellent according to label, and products that include higher percentages of DEET would be most recommended, and using permethrin on clothing. Altering behavior to minimize your potential for coming into tick habitat. So, minimizing contact with wooded and brushy areas, leaf litter, things like that. And, more importantly, when coming in from outdoors, from being in potential tick habitat or participating in activities where you might have been outside, it's really important to just, to check you and your family thoroughly for ticks. As I believe has been mentioned during this presentation a few times, the transmission of Lyme disease does take at least 24 to 48 hours for transmission to occur after a tick has been attached. So, prompt removal of ticks can help minimize risk of Lyme disease. So, CDC is actively working on development of newer prevention methods and as well as evaluation of prevention methods. But as we highlighted in our talk, the expansion of where disease is occurring is growing, and really we need to identify some options that are both highly effective at preventing disease, but also acceptable so that the majority of the U.S. population can use them and will use them.
Thank you. And let me just ask if anyone else, Ellie, would you or Alan like to add to that response at all?
Well, this is Alan. I think it may be worth commenting about the new repellents that have come on the market and over the last 10 years, the most recent which is Picardin, which, some of these have been used in Europe for a long time with good success, and I think a lot of people are a little nervous about DEET.
Okay, thanks. And so there's a question here from Jennifer Lee, and this is I guess, again, we can put this forward to whoever would like to answer this. But the question is, what are the DEET recommendations for pediatrics in regards to tick prevention? And is this the best approach? So that's a good follow-up to Alan's comments. Would someone like to volunteer to take that question?
Kirsten, I think I would just say that CDC isn't responsible for the licensing of what the labeling is for repellent use. So I think it's important for people to read labels appropriately, understand the the limitations and what has been evaluated and what hasn't, and in what different age groups.
Yeah, I would agree, and I would just really emphasize that repellents and insecticides in general, they should be used according to the labels, and that's why the label's there. And actually, EPA has done a lot of work recently on improving the labels that are on repellents in particular, and coming out with symbols and specific recommendations for their use. So I would reiterate what Dr. Kugler just said.
And just to add on to that, this is Ellie Thiel. The EPA actually has a tool on their website for which insect repellent is most appropriate to use, depending on how long you're going to be outside, kind of what region you're going to be in, or what season you're going to be in. So people can go to the EPA website and kind of input all of that information to get specific recommendations for what insect repellent to use.
Okay, thank you. Here's a question from John Skolstad, and the question is this: do nymphal ticks swell after being on humans for the 24 to 48 hour time period? Are they significantly more visible? And so can toss that out to any particular one of our speakers.
This is Becky. I can take that one. So typically when the nymphs will attach to you, they'll spend a little bit of time kind of getting their mouth parts into the skin and preparing to feed. And so during the first 24 hours, you're not going to see a big change. But that big change will occur around 36 hours, which we call sort of the big step, which is also when transmission is most likely to occur. So they will become more visible after they've fed.
Thanks. Do any of the other speakers, would they like to elaborate on that at all, or is that good? Okay, so I'm not seeing any other questions that have been asked. I'll give it just a moment longer. So maybe one that's coming in here.
Well, I see there is a question about Borrelia mayonii.
Okay, good.
From Jeff.
Oh, yes, I see it. So the question is, given the elevated spirochetemia for the new species, would we expect to see spirochetes on stained blood smear? And we did actually see spirochetes on Giemsa stain for, I believe, 1 or 2 of the patients, so it is possible.
Good, thank you. Okay, well, with that, I want to thank everyone for their participation today. And so if, as I mentioned before, the webinar will be made publicly available once we have the transcripts done and get it posted up online at the website. So I thank you for your participation today and wish all of you a nice Friday and a nice weekend.
Thank you.
That does conclude today's call. Thank you all for participating. You may disconnect your lines at this time.